Good day. So this is the first part of our lecture discussion for the nervous system. The two main divisions of the nervous system are the following. First, you have the central nervous system or CNS. It includes the brain and your spinal cord. The second one is the peripheral nervous system or PNS, and it includes all neurons outside of the central nervous system, including the nerves of the face, your ganglia, nerves of the upper and the lower limbs. The different functions of the nervous system are as follows. First, receiving of sensory input. Second, integrating information. Third, control of your muscles and glands. Fourth, maintaining your homeostasis. Fifth, establishing and maintaining mental activity. Apart from having the main systems under the nervous system, we also have different divisions. The first division would be your sensory division. It conducts action potentials from sensory receptors to your central nervous system. The second one would be your motor division. It conducts action potentials to effector organs such as muscles and your glands. You also have your somatic nervous system. This system transmits action potentials from the CNS to your skeletal muscles. You also have autonomic nervous system, which transmit action potentials from the CNS to cardiac muscles, smooth muscles, and glands. And lastly, you have your enteric nervous system, which is a special nervous system found only in the digestive tract. So this diagram shows you the main divisions of the nervous system and how they interact with each other. So the sensory division starts with a sensory input which can be brought about by the exposure to light, sound, taste, smell, touch, temperature, pain, or pressure. Then the action potentials are being sent by your sensory receptors to your sensory division, and then eventually going to the central nervous system, wherein your brain and your spinal cord decodes the message brought about by the action potentials. So, with the response, it is being sent to your motor division. The response can be a somatic response, where your effectors are your skeletal muscles, or it can also be an autonomic response, which can be a sympathetic or a parasympathetic response, and the affected muscles in your body are the cardiac and smooth muscles as well as your glands, and the movement or the motor output can be movement, changes in metabolism, heart rate, or breathing rate. The cells of the nervous system include your neurons. They actually receive stimuli and conduct action potentials and transmit signals to the other neurons or effector organs in your body. Next, you have your glial cells. They are supportive cells of the central nervous system and the peripheral nervous system, meaning these cells do not conduct action potentials. Instead, glial cells carry out different functions that enhance the neuron function and maintain normal conditions within the nervous tissue. The neuron or nerve cell structure. So for your neuron, what you first see is of course the neuron cell body. So the neuron cell body is actually the one processing the stimulus, and it contains the nucleus of the nerve cell. It also has what you call your dendrites, which receives your stimulus from other neurons or sensory receptors. And posteriorly to the cell body, it actually shows you now your axon. So this transmits stimulus to the gland, muscle, organ, or other neuron. There are different structural types of your neurons. First, you have your multipolar neuron, which has many dendrites and a single axon. Most of them are found in your central nervous system and most of your motor neurons. Next, you have your bipolar neuron, which has one dendrite and one axon, and they are found in your eye and nasal cavity. Lastly, you have your pseudo-unipolar neuron, which has one axon and they don't have any dendrites, and most of them are actually sensory neurons. You also have what you call your glial cells. These are the supportive cells of the central and the peripheral nervous system, and they are actually more numerous than the neurons. They can divide to produce more cells, and we have 5 types. The first one, you have your astrocytes. They are star-shaped. They are most abundant and they form the blood-brain barrier. Next, you have your ependymal cells. They produce and circulate cerebrospinal fluid or the CSF in your brain. Next, you have your microglia. It helps remove bacteria and cell debris from your central nervous system. You also have your oligodendrocytes, which produce myelin sheath in the CNS. And lastly, we have your Schwann cells, which produce myelin sheath in your peripheral nervous system. This table summarizes the different cells found in the nervous system, may it be a neuron, a glial cell that can be found in our central and nervous and peripheral nervous system. They do have the description here and of course the functions that you should know when it comes to identifying these different nervous system cells. So this summary can also be seen or can also be found in your book, and of course a copy of the lecture that will be provided to you. Now let's talk about the myelin sheath. So myelin sheaths are specialized layers that wrap around the axons of some neurons, and those neurons are termed myelinated. So again, once a neuron does have myelin sheath, you term it as myelinated neurons. So these sheaths are formed by the oligodendrocytes in the central nervous system and Schwann cells now on your PNS. So myelin is an excellent insulator that prevents almost all ion movement across the cell membrane, and the gaps that you see in the myelin sheath are what you call your nodes of Ranvier. This occur about every millimeter where ion movement can also occur. There is what you call the saltatory conduction or the jumping of action potentials in the axon, and myelination of an axon increases the speed and efficiency of action potential generation by about 3 to 15 meters per second than unmyelinated axons. Multiple sclerosis is a disease of the myelin sheath that causes loss of your muscle function. So if there's a disease of myelin sheath, it would actually cause you multiple sclerosis. So for unmyelinated neurons, they are axons that lack the myelin sheaths. These axons rest in the indentations of the oligodendrocytes in the central nervous system and the Schwann cells in the PNS. A typical small nerve, which consists of an axon of multiple neurons, usually contain more unmyelinated axons than the myelinated axons. So to compare your myelinated axon and unmyelinated axon, for your myelinated axon, it has two Schwann cells forming a part of the myelin sheath around each single axon, and each Schwann cell surrounds part of one axon. So as you see here, the nucleus is found peripherally, and then at the center you will see there the axon, and it is being surrounded by your myelin sheath. But with your unmyelinated axon, it has or it is with two Schwann cells surrounding several axons in parallel formation, and each Schwann cell surrounds parts of several axons. So in this one, you will see several axons around it, and the nucleus is found centrally. So to compare, for the CNS, you have your oligodendrocytes to produce the myelin sheath, but for your PNS, the myelin sheath are being produced by your Schwann cells. And then for the collection of axons, you have your nerve tracts for your central nervous system, and you have your nerves in your PNS. And then for the collection of cell bodies, you have nuclei for your central nervous system, and you have ganglion for your peripheral nervous system. The nervous tissue varies in color due to the abundance or absence of myelinated axons. So nervous tissue exists as gray matter and white matter. The gray matter consists of groups of neuron cell bodies and their dendrites where there is very little myelin. On the other hand, white matter consists of bundles of parallel axons with their myelin sheaths, which are whitish in color. So if you observe the picture that you're seeing here on your screen right now, you see that some parts of your brain or some parts of your spinal cord do actually have gray color on them or white color in them. So basically, the color of the parts of your brain or spinal cord would actually let you know that the gray matter is a collection of dendrites and the cell bodies, while the white matter is a collection of axons and their myelin sheath. So in imaging techniques, you will also be able to see how your gray and your white matter look like. So for electrical signals and neural pathways, so first we should know what a resting membrane potential is. So in your neurons, usually you have what you call your resting membrane potential and your action potential. So when you say resting membrane potentials, hence resting, of course, your neurons are at rest. But when you say there's an action potential acted by it, there's already the presence of stimulus. Okay, that should um, be processed in your system. So these potentials are mainly due to the difference in the concentration of ions across the membrane channels, and the sodium-potassium pump. So the membrane channels include your leak and your gated channels, right? So before we go on to the differences of your leak and gated ion channels, remember that when there is a resting membrane potential, outside of your cell is more sodium rich, and then inside of your cell is more on potassium rich, like what we have discussed in the muscular system, right? That when your muscles are rested, more of the potassium are inside your muscular system, and outside of your muscles, you have more of the sodium. But once the sodium already comes in your muscles, that's when contraction happens. So that is also the same sequence that happens with your nervous cells or nerve cells or nervous system. Okay, so again, the differences of your leak and gated ions, when you say leak channels, these are always open. So these channels can let um, your your ions come in and out of the cell. But when you say gated ion channels, they are um, generally they are closed, but it can be open due to the influx of voltage that can be brought about by your sodium ions, or also the presence of some chemicals like your neurotransmitters, just like in the muscle contraction, like your acetylcholine. Okay, so the leak membrane channels are always open, and ions can leak across the membrane down their concentration gradient. Because there are 50 to 100 times more potassium leak channels than sodium leak channels, the resting membrane has much greater permeability to the potassium than to sodium. Therefore, the potassium leak channels have the greatest contribution to the resting membrane potential. So with the gated membrane channels, they are closed until opened by specific signals. Chemically, gated channels are opened by neurotransmitters or other chemicals, whereas voltage-gated channels are opened by changing in the membrane potential. When opened, the gated channels can change the membrane potential and are thus responsible for the action potential. So with the sodium-potassium pump, it compensates for the constant leakage of ions through your leak channels. The sodium-potassium pump is required to maintain the greater concentration of sodium outside the cell membrane and potassium inside. The pump actively transports sodium into the cell and sodium out of the cell. It is estimated that the sodium-potassium pump consumes around 25% of all the ATP in a typical cell and 70% of the ATP in a neuron. So for the resting membrane potential, sodium tends to diffuse into the cell and potassium tends to diffuse out. In order to maintain the resting membrane potential, the sodium-potassium pump recreates the sodium and potassium ion gradient by pumping sodium out of the cell and potassium into the cell. So the resting membrane potential exists because of the following conditions: the concentration of potassium being higher on the inside of the cell membrane, and the concentration of sodium being higher on the outside. And then the presence of many negatively charged molecules such as proteins inside the cell that are too large to exit the cell. Lastly, the presence of leak protein channels in the membrane that are more permeable to potassium than it is to sodium. So this is actually how the sodium-potassium pump happens when in terms of us discussing about the nervous system. Okay, so again, there are three conditions wherein there is a resting membrane. First is that kapag ka ang sodium ay mas mataas sa labas ng cell compared sa loob. So, ibig sabihin, mas mataas yung concentration ng sodium sa labas, and then sa loob niya, mas madaming potassium. Again, kapag ka mas maraming potassium, more likely your muscles or your nerve cells are in or at rest. Okay? The second condition is that kung meron pang ibang types of ions or may iba pang mga proteins within the cell na hindi kayang lumabas through your leak channels. Okay? And then lastly, another condition is that dahil mas marami nga ang leak channels ng potassium kaysa sa sodium. So, kumbaga, mas maraming kayang pumasok na potassium sa loob ng cell anytime because of these leak channels. Okay? So, again, with the sodium-potassium pump, just always remember C3 na antukin. Okay? So, always the concentration gradient kapag ka naman nagkakaroon na ng action potential is nagbabaliktad. Pag may action potential, dapat mas mataas yung potassium niya sa labas and then sa loob mas marami yung sodium. Okay? But then again, it will depend on the amount of neurotransmitters or other ions that could actually affect the opening of your voltage-gated channel and how many or how much voltage can be that can get into your cell para magkaroon ng action potential. Okay, so the concept of your neurons on how they work is just is like how much they also work with your muscles. Okay. So the action potentials, these are actually what you call the electricity that cause the depolarization and repolarization of your neurons. So the change resting membrane potential is by activating the gated ion channels, and we have what you call your local current. This is the movement of the sodium ions which causes inside of the cell to be more positive, which actually ignites the depolarization. Okay, again, please remember how your depolarization and then repolarization and then resting membrane happens. Okay, so the more electricity or the more sodium that comes in, your cell becomes more positive, and of course it ignites or triggers the depolarization. Okay. So, action potentials allow conductivity along nerve or muscle membrane and similar to electricity going along an electric wire. The channels responsible for the action potentials are voltage-gated sodium and potassium channels, which are closed during resting membrane potential or during rest. Okay, so it is more likely that what we need in igniting the movement of your neurons is that it's because of the voltage. Okay, so when a stimulus is applied to the nerve cell following neurotransmitter activation of chemically gated channels, sodium channels open very briefly and sodium diffuses quickly into the cell. So if enough sodium enters, then there will be a threshold. Again, when you have already reached a threshold and more sodium channels open, once threshold is reached, there should be an all or none law that applies. Okay, like your muscles, the movement of your or the reaction of your neurons is does actually apply the all or none law. Okay, hindi po pwedeng isang neuron lang ang pipitik dyan, kailangan lahat sila. Okay? Kasi kung isa lang yan, of course, hindi magkakaroon ng motor movement. Okay? Siyempre, wala naman kayong nakita na kunwari sa kuryente, na isa lang ang gumagana. Usually, kailangan lahat ng linya ng kuryente ay gumagana. Okay? So, yun yung iisipin nyo na concept pagdating naman sa action potentials na nangyayari sa mga neurons. Okay? Kung ang linyada ng isang kuryente ay iisa lang, dapat lahat ng linyada na yan gagalaw. Okay? Pag may threshold, pagka na-reach na yung point na yun, of course, yun na mag-umpisa yung depolarization because of the action potentials. And then action potentials will continue until sodium channels close and then potassium channels open. Once they open, of course, papasok na naman yung mga potassium ions and then repolarization occurs or going to rest naman na ulit yung mangyayari para sa response ng inyong nerve cells. And then the sodium-potassium pump restores. Okay? So again, this is actually how it looks like for action potentials in your nervous system. So again, around negative 70 millivolts, that's the ideal resting membrane potential. And then, of course, the local potential. And then you have your threshold. And then, of course, once there's already enough or so much influx of your sodium, of course, you will see their depolarization. And then, around around positive 20 millivolts, that's already the peak of its action potential. And then, once this action potential has already been reached, of course, again, ito na yung sinasabi na magko-close na yung mga sodium channels and then your potassium channels will reopen, and of course, the influx of the potassium would succeed, so repolarization now happens. And then going to hyperpolarization, that's below the resting membrane, and then eventually going to its resting membrane potential again. Okay? So this full potential or action potentials for your nervous system would actually occur around 1 or 2 milliseconds. So ganun siya kabilis. Okay, imagine how fast your voltage-gated channels open and close and then the influx of your ion happens. So ganun kabilis ang process ng action potential when it comes to your nervous system. With the voltage-gated ion channels and the action potentials, this is how it goes. No? Para i-explain ko sa inyo ng mas maayos. So again, when resting membrane, ibig sabihin at rest, okay? Ang makikita natin dyan is more of your sodium ions are outside of your cell. Okay? And then you see here that your potassium channels are actually closed. Okay? So it's around negative 70 millivolts. That's the resting membrane potential of your nerve cell. Okay? So next you have your depolarization. Okay? With the depolarization, you will see here that from negative 70 millivolts, it would actually pass through the threshold. You have your local potential and you will be reaching the depolarization at around the maximum of positive 20 millivolts. So in this step, you will see that your sodium channels open here, and then of course the influx of the sodium goes in of the cell. Okay? And then what happens is that for repolarization, the sodium channels close and then the potassium diffuses outside of the cell, okay, causing it for the process of repolarization going to your hyporepolarization and then of course going back to its resting membrane potential. Okay? So for unmyelinated and myelinated action potentials. So action potentials are conducted slowly in unmyelinated axons and more rapidly in your myelinated axons. Action potentials along unmyelinated axons occur along the entire membrane, and action potentials on myelinated axons occur in a jumping pattern at the nodes of Ranvier. So this type of action potential conduction is again what you call your saltatory conduction. So in unmyelinated axon, it looks like this. So it is continuous. Okay. So outside of the membrane becomes negative and then the inside of the membrane becomes positive. So that's the part wherein it is on the depolarization side or the resting membrane. And then, so for myelinated axon, instead of going all throughout the membrane, what happens is that it's jumping from one node to another node. Okay, so if this is your internodes, you have here the node of Ranvier, you have the Schwann cell in between. Okay, so instead of the action potential traveling all throughout, what happens is that they jump from one node to another. So it's faster rather than having your unmyelinated axon. So for the axon conduction speed, the speed of the action potential conduction varies widely, even among myelinated axons. It is based on the diameter of the axon fibers. Medium diameter, lightly myelinated axons, characteristics of autonomic neurons, conduct action potentials at the rate of about 3 to 15 meters per second, and the large mid-diameter heavily myelinated axons conduct action potentials at the rate of 15 to 120 meters per second. So next is a neuronal synapse. So it is a junction between where the axon of one neuron interacts with another neuron. The end of the axon forms a presynaptic terminal, and the membrane of the next neuron forms the postsynaptic membrane with a synaptic cleft between the two membranes. So the chemical substances called neurotransmitters are stored in the synaptic vesicles in the presynaptic terminal. So, like your neuromuscular junction, of course, the synapses are like the spaces in between. Okay, so these are where your axon attaches to your muscle, gland, organ, or other neurons. And again, it involves your neurotransmitters. So, one action potential reaching the presynaptic terminal causes voltage-gated calcium channels to open, and calcium moves into the cell. This influx of calcium causes the release of neurotransmitters by the exocytosis of the presynaptic terminal. The neurotransmitters diffuse across the synaptic cleft and bind to the specific receptor molecules on the postsynaptic membrane. The binding of neurotransmitters to these membrane receptors causes chemically gated channels for sodium, potassium, or chloride to open or close in the postsynaptic membrane. The specific channel type and whether or not the channel opens or closes depend on the type of neurotransmitter in the presynaptic terminal and the type of receptors in the postsynaptic membrane. The response may either be stimulation or inhibition of an action potential in the postsynaptic cell. If sodium channels open, the postsynaptic cell becomes depolarized and an action potential will result if threshold is reached. If potassium or chloride channels open, the inside of the postsynaptic cell tends to become more negative or hyperpolarized, and an action potential is inhibited from occurring. Again, this is actually how your action potentials affects the release of ions in the presynaptic terminal. Again, once action potentials has already been received from the brain or your spinal cord, your calcium channels actually opens, and then of course it gives you also the influx of calcium inside of your presynaptic terminal. So in this presynaptic terminal, you will see your synaptic vesicles that when they are released, okay, or through exocytosis, they release the neurotransmitters that are bound to the receptor sites. Okay, these are the receptor sites that opens, and then it allows the influx of your sodium going to the cell in the postsynaptic membrane. Okay, just like your neuromuscular junction, this is actually how your receptor sites open. Okay. So, neurotransmitters are a type of chemical messenger which transmit signals across your chemical synapse. There are many neurotransmitters, with the best known being your acetylcholine and norepinephrine. Neurotransmitters do not normally remain in the synaptic cleft indefinitely, thus their effects are short duration. These substances become reduced in concentration when they are either rapidly broken down by enzymes within the synaptic cleft or are transported back into the presynaptic terminal. An enzyme called acetylcholinesterase breaks down with the acetylcholine, and the norepinephrine is either actively transported back into the presynaptic terminal or broken down by enzymes. So this table here shows you the different neurotransmitters that are found in your nervous system. So you have here your acetylcholine, and you see here the site of release, like in your CNS synapses, ANS synapses, and the neuromuscular junction. Its effect is excitatory or inhibitory, and some clinical examples have been provided here, like if you have problems with acetylcholine production, you can develop Alzheimer's disease. So please read up on this table and make sure to be familiarized with the clinical examples that are or that can cause the diseases regarding with the neurotransmitters that have been provided in this table. Okay, so for example, if you don't have dopamine in your body, what type of clinical disease could you actually acquire with this one? Okay. So reflexes. So a reflex is an involuntary reaction in response to a stimulus applied to the periphery and transmitted to the central nervous system. So your reflexes allow a person to react to stimuli more quickly than is possible if conscious thought is involved. So the components of the reflex arc include the following. First, you have your sensory receptors. This pick up the stimulus like your skin. Next, you have your sensory or afferent neurons, which send stimulus to the interneurons in the spinal cord. And then you have your interneurons or association neurons, which are located in the central nervous system and connects to the motor neurons to process your stimulus. You also have your efferent or motor neurons that send response to the effector, and of course your effector, which are your muscles, glands, or organ. So this is the reflex arc. So again, you have here first your sensory receptor like your skin. So if action potentials or stimulus are being sent from the skin, it would go to your sensory neuron, and then to a ganglion, and then to the interneuron in your central nervous system. And then the action potentials will be sent back to your motor neurons, and then eventually to a nerve, and then to your effector organ, which can be your skeletal muscle. So that's how your reflex arc look like. So the CNS has simple to complex neuronal pathways. First, you have a converging pathway, which is a simple pathway in which two or more neurons synapse with the same postsynaptic neuron. This allows information transmitted in more than one neuronal pathway to converge into a single pathway. And you also have what you call your diverging pathway, which is a simple pathway in which an axon from one neuron divides and synapse with more than one other postsynaptic neurons. This now allows information transmitted in one neuronal pathway to diverge into two or more pathways. So this is actually how your converging and diverging pathways look like. Okay. So next we have the idea of summation. So this is a single presynaptic action potential usually does not cause a sufficiently large postsynaptic local potential to reach the threshold and produce an action potential in the target cell. So many presynaptic action potentials are needed in a process called summation. The summation of signals in neuronal pathways allows integration of multiple sub-threshold local potentials, and summation of the local potentials can bring the membrane potential to threshold and trigger an action potentials. So when you say summation, ito yung parang hindi naman talaga siya super umaabot ng threshold, pero sunod-sunod siya. Okay, para siyang kumbaga actually pinakita ko na before kung ano itsura ng graph ng summation, di ba? Para siyang tuloy-tuloy, and then it will repolarize, and then tuloy-tuloy siya. Hindi siya katulad ng action potential na, or full action potential na it will reach up until it reaches the voltage that it needs before it repolarized. Okay, so summation sunod-sunod siya. Okay, sunod-sunod siya na action potential, and then may isang hanggang ma-reach na yung pagre-repolarize. Okay? And then you also have what you call your spatial summation, which occurs when the local potentials originate from different locations on the postsynaptic neuron, for example, from your converging pathways. You also have your temporal summation, which occurs when the local potentials overlap in time. And this can occur from a single input that fires rapidly, which allows the resulting local potentials to overlap briefly. And then spatial and temporal summation can lead to stimulation or inhibition depending on the type of signal being produced. So that ends the first part of our nervous system lecture. Please watch out for the second video for the continuation of this discussion. Thank you.